And good morning, everyone. Good afternoon, whatever. I never know how to start these things. So, we're going to finish up on Chapter 7, our control of microbial growth. We've been talking about some of the physical means, things like temperature, both hot and cold, things like pasteurization, which, got us into the terms of sterilization versus sanitation. Sterilizing means everything's dead. Sanitation means the number of bacteria have been reduced to a level that they won't present any kind of health problem. But if you let it sit long enough, it will begin to grow bacteria. And for foods and things, it begins to spoil. We also talked about some of the other physical mechanisms, things like soap, which probably could be actually a chemical means, but regardless, certainly the idea of sterile filtration. So we can have filtered air, which is a really wonderful way to reduce bacteria load. UV light and radiation are often used, UV light particularly in conjunction with sterile air in sterile cabinets, what we call a laminar flow hood, where we can begin to reduce the bacterial load to essentially nothing, and you can have a sterile air environment. We also then kind of hit on some of the other or microbial reduction techniques that are often associated with food, things like cooking, refrigeration, and the ability to pull water out of the food and the bacteria to make it so that it's not very habitable, it's not a very pleasant place for the bacteria to grow, and they may be there, but they're not going to grow. And so this would be things like salting or even sugaring. When you increase the osmotic pressure, another way of saying you've got a whole lot of material of salts or sugars that are outside, the cell begins to lose water. Water always follows salt. And so we can dehydrate material, and that will very much keep the bacteria from growing on it very well. So we now turn our attention to what we think of as chemical means. Now, another point we talked about when we're dealing in the healthcare biz is that we have two different kinds of things we might be trying to clean. We have surfaces and instruments, solid materials, and we can do all kinds of fairly nasty things to these stuff like this metal. You can put it in an autoclave. Surfaces of operating rooms and hospital rooms and beds, they can be exposed to UV light, and that'll do a pretty good job. We have air filtration kinds of systems. When you're talking about the individual, that becomes a lot more difficult because the sterilants that we would use on instruments are going to be toxic, or temperatures and conditions that are so harmful that living organisms can't survive, including us. So you can't sort of put yourself in an autoclave. You can't incinerate the outer layer of skin. You might be able to do that, but that'd be kind of difficult to do. So it's some things to think about. Now, with chemical means, we have a couple of different kinds, again, terms. The idea of a sterilant. Here, let me get my little red waving dot to whatever extent that works. I like it anyway. We have the idea of a sterilant. Now, this is, again, sort of the term sterilizing versus sanitizing. A sterilant is absolutely going to kill everything, all actual or potential life. That means endospores, bacteria. It's stuff that's going to be pretty darn toxic. And things like this would be ethylene epoxide, which is probably used industrially. Glutaraldehyde is a fixative, very much like formaldehyde. These are things that are going to absolutely sterilize surfaces or materials, and so they actually work fairly well. They are quite toxic, though, so you can't really use them on people. Disinfectants are kind of like a sanitizing agent, and we can think about these in our own homes. We have disinfectants that are going to be lowering the bacterial numbers. They probably, a disinfectant, if it works really well, is probably going to be a little hard on your skin. So washing your skin with bleach, not a good idea. Hydrogen peroxide actually is a brilliant disinfecting agent. It actually will break down to generate water and the oxygen radical, which we've talked about. The oxygen radical is extremely oxidizing. It picks up electrons and it's going to pull electrons from things. It is in concentrations that are going to be useful for disinfectant, maybe a little bit too strong to actually use internally or to take on the surface. We also have, of course, hydrogen peroxide that is diluted to about a 2 or 3, 5% strength, and that is used for treating wounds. You can gargle with it, and it's a little funky. It will bubble up, and it'll actually start to bleach stuff, but it's not anything too serious. Across the hall in the lab, I've got 20% hydrogen peroxide, and that stuff is actually really quite corrosive. One of the things to think about, just in the overall biz of operating in and trying to develop sterile techniques and things, is that things like bleach, and particularly hydrogen peroxide, they've got a finite shelf life. So you can have a jug of bleach, Clorox, that if you've let it sit under the sink for a year, it'll smell pretty chlorine-y and pretty funky, but it doesn't have a whole lot of bleaching capacity anymore. It actually breaks down. Hydrogen peroxide definitely does. Hydrogen peroxide, if you see in the grocery store, is sold in those brown bottles. That's because light will actually increase the rate of breakdown, so you have to keep it out of the light. But even still, over time, hydrogen peroxide starts to lose its oomph, and so they sell it in small bottles, which is a good idea, so you can use it and buy another bottle, rather than trying to store it in a large container. We have things that we refer to as antiseptics, and antiseptics are a term we would use for something that is working as a disinfectant, that is, reducing the microbial numbers, but it's now something that you can use on skin. Probably toxic if it's taken internally, but excellent examples would be things like alcohol. So when you get ready to inject somebody, you're going to start an IV, you're going to rub them down with a alcohol pad. What that's doing is reducing the bacterial load in that specific area, and it's not particularly dangerous or toxic to the skin, and you can then have a reasonably sterile environment in which you're going to jab somebody. Other kinds of chemicals that you may not necessarily know of, benzalkonium chloride. This is what's in Bactine. It's also in things like Physohex, these sterilizing soaps and things. And I'm trying to think of one. Physohex is one of them. There are a variety of soaps that when you're preparing for surgery, like if you have elective surgery, you're going in and they're going to open you up, doctor might actually have the patient shower with one of these kinds of antimicrobial soaps, and it's just again to reduce the bacterial load on the skin surface. So they work pretty well. You have to be careful with them. The other side of this is you remove the bacteria on your skin, and you are opening up your skin for other kinds of infections. It's not necessarily something you want to do a whole lot, but they certainly can reduce the load. Of course, a classic chemical means a class of drugs we know as antibiotics. And antibiotics are very specifically inhibiting the growth of bacteria. Now, this works because bacteria, while they are phenomenally similar to us in all other biochemical mechanisms, as we've been talking about with glycolysis and things. Bacteria break down sugar the exact same way you do. Bacteria have DNA. They use the same code for the genetic code for translating RNA into proteins, but there are some interesting and significant differences in some of the enzymes and structures that are involved in that kind of gene mechanisms, transcription and translation, and that difference can be exploited. Additionally, there are things that bacteria do, like the presence of a cell wall, that we don't have. And so there are a range of different antibiotics that have been identified that are able to inhibit the growth of bacteria, and they are generally produced by other microorganisms, either fungi. For penicillium is a mold that you can actually culture quite effectively in your refrigerator on different bits of fruit. A whole range of soil both fungi and bacteria produce antibiotics that are generally non-toxic to eukaryotes or to complex organisms such as ourselves, but are quite toxic to the bacteria. So just, not inclusive, but penicillin, streptomycin, cephalosporin. We'll talk a little bit about antibiotic resistance as we get further into some of the genetics, because that's actually a really cool idea in the area. But antibiotics are constantly being developed. Antibiotics are being tweaked, and they have side effects for sure. One thing is that taking an internal antibiotic, because an antibiotic would work on the surface too, of course, but taking antibiotics internally can cause changes to the gut microflora, and that's going to lead to various kinds of digestive issues. And people can have very dramatic allergies to these things, and a range of different kinds of phenomena can happen when you take these antibiotics. But they also are working very well topically. So you have things like various antibiotic creams that you put on a cut. Yeah. So let's see. I was just trying to think of what is the Neosporin, which has got neomycin in it, and a variety of different kinds of antibiotics, usually like a triple whammy, and you can put that on a wound, and it's going to inhibit the bacterial growth there. So one of the things that is interesting is that we can classify the degree of toxicity by using something that is known as the phenol coefficient. Now, we talked about Professor Lister and his use of what he called carbolic acid, what we know in organic chemistry as phenol, which is the benzene ring, a 6-carbon ring structure with a hydroxyl or an OH group coming on it. Phenol is a very toxic material, so it can't be taken internally. It is really good at sterilizing stuff. And so we can use this as kind of a baseline of how well something works to kill off bacteria. And so this is the phenol coefficient. What we're doing is measuring the killing capacity of a certain compound in relationship or relative to phenol. So if you have a compound that has a phenol coefficient greater than 1, like that, it means it's more toxic than phenol. If it's less than 1, it's less toxic. So phenol is not the end all. There are things that are more toxic than phenol. It just is a good sort of baseline measurement of how well things work. So the way we would do this is look at the minimum concentration that would inhibit growth. And we have different kinds of things. There's the 5-minute exposure and then the 10-minute exposure. That is how long it takes for this thing to actually kill stuff. And then you can test a disinfectant and compare it. And so in this case, we've got a 1 over the test with the lowest no growth at 10 minutes, not at 5 minutes, over 1 over the phenol lowest no growth at 10 minutes, not at 5 minutes. And in this specific example, we end up with a phenol coefficient of 2. So greater than 1 means it's more toxic to the bugs than phenol. Less than 1, it is less toxic. And so this is something that is going to be actually more toxic. And you can just sort of eyeball it here, is that the disinfectant is killing stuff at a lower concentration. These are a bunch of different dilutions of the disinfectant than the phenol is. So that's a way of standardizing different kinds of disinfecting agents. So there are a number of different modes of action that we can kind of cluster into some different kinds of in sort of categories. We've got things that act as protein denaturants. That is, they're going to break apart the secondary and tertiary structure. Now we know that from a physical mechanism, heat is a brilliant way to do this. But chemicals can also do this as well. These compounds that contain reactive halogens, things like iodine or chlorine are the primary ones. Bromine is another halogen. Bromine is quite toxic and not very used a lot. These are chemicals that will, when they begin to decompose, will begin generating these different kinds of halogens that are very reactive. And so you can use chlorine tablets. Iodine is another halogen, and so iodine tablets and iodine solutions, these work. So here's an example, just as we're talking about with Clorox, sodium hypochlorite bleach. And because this is so reactive, both the pills and the liquid have a finite shelf life. With the pills, what happens is they start to absorb water and they begin to break down in the jar. One of the things you often see with a old bottle of chlorine tabs or iodine tabs is the lid starts to get really rusty. It starts to corrode. Probably not the best to have in your first aid kit, or you need to cycle it out. And bleach probably should be tossed out after a few months or so. So phenols, they're able to get into the protein structure. The halogens are going to be oxidizing everything, including proteins. Phenols are getting into the protein structure, sort of wedging themselves in and disrupting the secondary and tertiary structure. There are heavy metals that are often used, things like mercury compounds. And something that's not used so much, but this compound called mercurochrome, back in the day, in the 50s, this was a standard treatment as an antibiotic on a wound. Mercurochrome used to sting like a son of a gun, though. Copper salts, like cupric sulfate, is another thing. Silver nitrate is often, maybe even still used as as some kind of bacterial disinfectant. You can dip silver nitrate into wounds, and it also will then begin to break apart different kinds of protein structures. Thimerosal has, for a long time, been used as a preservative in everything from vaccines to contact lens cleaner. And in the past 15, 20 years, thimerosal has kind of come out of favor because it does have a little bit of mercury, which can lead to some toxicity. The way, the amounts that are present in different kinds of solutions is vanishingly low because it's very effective, and so by the time you inject somebody with it and you dilute it out, it's not really all that significant as a toxin. Still, it's probably not the best thing to be using. So most compounds now advertise themselves as being thimerosal free, and they have removed thimerosal and used other things that may or may not be any less toxic. It's just they're not carrying mercury. All right, so other classes of compounds that are useful are things that are going to begin not interacting with proteins, but disrupting the bacterial membrane. And so this is where we can get into things like soaps. Soaps do a really good job of emulsifying or breaking apart the plasma membrane. So the detergent molecules kind of get into the plasma membrane and they begin to cause it to open up, and then water can get in there and blow the bacteria apart. So soap does a very good job of breaking apart bacteria. Alcohols also do a really good job of entering into the plasma membrane and causing disruption. They do a whole range of different things. They can pull water out. They can break apart the membrane. One of the things that's interesting, we're using a lot of hand sanitizers these days, and occasionally you'll read about hand sanitizers that are not safe, in part because you put hand sanitizer on your hands and then you eat. And you can be transmitting material from your hand sanitizer onto your food. Hand sanitizers pretty much are using alcohol with a 60% concentration or greater. So you can't use vodka, which is about a 50% or 100 proof. You got to have something that's a little bit higher than that. So 151 rum, that would work really well. And one of the things that has been done, and I'm always kind of sniffing my hand sanitizers just to see what's going on with them. Most hand sanitizers are made with ethanol because it's fairly cheap to generate. It works really well, and it's non-toxic. I mean, this is ethanol is what you're drinking when you're drinking vodka. But in order to get around having a liquor tax, you have to add something that will make it so it's non-palatable. This is referred to as a denaturant. And so you may pick up a bottle of rubbing alcohol of ethanol from the first aid aisle at Walgreens, and this doesn't have a liquor stamp on it, but it's actually the same concentration as Everclear or a really good rum. And it will be known as SD alcohol, which is standard denatured alcohol. And there's a recipe called SD1, which is a way of making the alcohol non-palatable. Basically, you add a chemical that is non-toxic but is very, very bitter. And I'm trying to think what it is. I can't think of the name of it, which is irrelevant. But it is like one of the most bitter flavors known to the human palate. And it makes it so you really can't drink rubbing alcohol very well. Now, there are other denaturants that we use in the lab. We've got industrial ethanol around here a lot that is not designed for healthcare purposes. It's not designed to be put on surfaces that could come in contact with the mouth. And they're denatured with things like methyl ethyl ketone, which is PVC pipe cleaner. They can be denatured with a little bit of methanol. They can be denatured with a variety of things. It makes it so not only they're non-palatable, they're also toxic. So that's an issue. As long as you're using standard denatured SD1 denatured alcohol, there's not a problem. I think actually we've got hand sanitizers around that are not even denatured. Several years ago, probably around the time of H1N1, so maybe like 10 years ago, in the Chicago prisons, they actually had to limit the amount of hand sanitizer they had in the facility because people were eating it and getting a slight buzz off of it. Well, whatever. I do say if you find yourself eating hand sanitizer just to kind of escape your troubles, you probably need to rethink your life a little bit. Still, I sniff hand sanitizer all the time and figure I'm walking with my cup of coffee and go by one of the big hand sanitizer pumps, just like put 2 shots in, like a little bit of vanilla syrup. It takes the edge off. No, I don't do that. All right. So there are other kinds of non-specific sanitizing agents, and these are things that are just general oxidizers, and they're going to oxidize everything. And so benzoyl peroxide is a standard compound used in disinfecting soaps, things like Physohex, we talked about, and Physoderm. They have benzoyl peroxide, which will oxidize a lot of materials on the surface of your skin, including the bacteria. Hydrogen peroxide, again, a brilliant oxidizer there. So we've got the molecule breaks apart to generate a free oxygen, that an O1 that is very, very corrosive. Ethylene oxide also generates the oxygen radical. Ethylene oxide is typically used as a gas in various chambers where you can put stuff in and sterilize them with ethylene oxide. So it's fairly benign to the material. Things like plastic can be sterilized with ethylene oxide very easily, whereas heat can't be used very well. So antibiotics, man, we could have an entire class or a course on the mode of action of antibiotics. And we'll be talking more about some of the antibiotic mechanisms as well. Antibiotics are chemicals, drugs that are going to block specific activities in the microbial cell. They're going to inhibit a range of different things. Now, antibiotics can be broken down into a variety of different classes. There are antibiotics that are going to block cell wall synthesis. And what that's going to do is make it so the bacteria are very sensitive to osmotic shock. They pop. And this is not going to kill the bacteria necessarily. What it's going to do is prevent them from replicating. Then there are inhibitors of protein synthesis. There are drugs like streptomycin, which actually can bind to the ribosome, that is a component of the protein translation machinery, and block proteins from being generated. So this may take a little bit of time as the bacteria are trying to generate new proteins to just operate. their protein translation machinery is being blocked. There are other kinds of antibiotics that can block the production of RNA, the RNA polymerase that is reading the DNA, and it can be blocked. No RNA is being produced, no protein is being produced, the bacteria is going to die. Another mechanism would be to interfere with DNA replication. So inhibiting replication is a great way to halt bacterial growth. If they can't divide, they can't spread. So we've got a whole range of different classes of antibiotics, and they do different things. And it's all dependent on the fact that eukaryotes, like you and I, have these subtle differences that make it so streptomycin doesn't interfere with our ribosomes. Now, at the same time, there are a variety of chemicals and small molecules in microorganisms that will affect eukaryotes, and these are real serious toxins. Things like the toxin from Clostridium botulinum, otherwise known as Botox, is a really strong inhibitor of neurons to pass the message back and forth, and so it'll cause muscles to no longer contract, which is why you can inject it in spots and you can have a relaxation of muscles. So if I really wanted to reduce the forehead wrinkles I have, you can inject Botox around here, and what you're doing is basically paralyzing the muscles. Not necessarily a great thing, but whatever. So we have to choose antibiotics that are specifically blocking bacterial mechanisms and not affecting eukaryotic mechanisms. Because, let's face it, in terms of killing stuff, it's a lot easier to kill all cells than it is to kill selective cells. A few more antibiotic examples. We've got penicillin, which is blocking peptidoglycan synthesis. And remember where peptidoglycan is? In the cell wall. And so it is going to particularly work on gram-positive bacteria, I believe, but it is affecting the cell wall production. And so I'm standing in the Leidy Tabor Science Building, and Professor John Leidy was working at Abbott Labs? No. Ofui in Indianapolis, whatever, where they were developing the antibiotic penicillin, and I think Professor Leidy's contribution was developing ways that you could stabilize penicillin. Apparently penicillin is, in its native state, is rather unstable, and he developed one of the mechanisms by which you can stabilize penicillin. So penicillin is going to block the ability of the cell, the bacterial cell, to form cell walls. And this is going to make the bacteria much more sensitive to the salt water concentrations in its environment and probably lead to the cell rupturing. And it's going to block the cell from being able to divide and continue replicating. And so you can see here on a plate, you have the mold Penicillium. This is a fungus. And we've got bacteria here, a Staphylococcus. There's this zone of inhibition that is an area where the penicillin is diffusing into the media and killing the bacteria in this specific spot. So this apparently is how penicillin was first discovered, was noticing that the mold actually inhibited bacterial growth. Now, this is one of the ways that antibiotics can be identified, is you take compounds, place them on a plate, and the plate has a bacteria spread on it, and you look for these halos or this zone of inhibition. And that's how you can know that, there's something in this microorganism, this fungus, or this chemical that is blocking the growth and development of these bacteria. Let's see, other antibiotics that perhaps you're familiar with. Erythromycin. This is inhibiting protein synthesis. It's blocking the ribosome from being able to actually move around and move itself down the RNA. So the ribosome and protein translation ends up being a fairly rich spot for antibiotic function, in that if we look here, Chloramphenicol can bind to a spot on the ribosome and block any further protein translation, so you can't get the amino acid chain can't transfer from this tRNA to this one. Tetracycline is blocking the ability of the ribosome to recognize the RNA sequences at specific spots. Erythromycin is blocking ribosome function. Streptomycin also inhibits this. All of these different kinds of phenomenon can often be overcome by single nucleotide changes in the gene sequence for the ribosome that can result in a single amino acid change, making it so the ribosome is no longer sensitive, and that's how you generate antibiotic resistance. Rifampicin is blocking RNA polymerase, and actually it's one of the more esoteric guys. Not that many antibiotics do that. Rifampicin is making it so the RNA polymerase is not able to actually interact with DNA and generate the RNA transcript. Pretty big molecule. In fact, a lot of these are very, very large, complicated organic molecules. So that is our discussion of the control of antibiotics. And we're going to be getting into some of the bacterial genetics and control of gene expression, which is a really cool area, and see where we can go from there. That's all I know. If you've waited through this presentation this long, thank you. I am working on grading exams. I'm going to have them do my best to get them back to you by the end of the week. I will do that, yes. And we'll see where we go from there. All right? Hey, thanks for your attention. Go wash your hands. I'm Dr. G. I'm out. Peace.